Okay, so good morning or afternoon, wherever you are in the world, everyone. Um, Welcome to this webinar with Life Science Innovation Network Japan. Just a couple housekeeping points. Please make sure that your mic is on mute, cameras are off, and if you have any questions, to put them in the chat box. And I'm just going to hand over to Masashi now, and I hope you enjoy the session.
Thank you very much. So a few words that Japanese government encourage a lot for the globalization for the Japanese startups. The government put like more than 6 billion pounds announced last year, and do a lot of collaboration in between Japan and the UK. And also we're speaking to British government and to encourage soft landing programs or management for the Japanese startups to allocate in this country. And I think this is a great opportunity to use this Genesis Innovation Workshop. My name is Masashi Matsunaga, CEO of Biospire, and also supporting LinkJ as a, how to say, like a acting ambassador, because I'm based in the UK. It's quite foggy, but my location is the Harwell campus near Oxford, and encouraging a lot of Japanese startups to use this facility, for example. Previously, in November, LinkJ member visited UK clusters, including One Nucleus, of course, and did a lot of great discussions. So we welcomed Soyama-san, the CEO of LinkJ, for a few words of introduction of LinkJ. Soyama-san, please.
Yes, Masashi-san, thank you very much for your introduction. Hello, Tokyo and UK. I'm Aki Soyama, CEO of LinkJ. LinkJ is a non-profit organization with its official name, Life Science Innovation Network Japan, L, in a wide range of life science, I, to support and promote innovation, and K, by creating network, or in other words, ecosystem, J, both in Japan and abroad. That is our mission. So we have about 750 members. Those LinkJ members and LinkJ itself have hosted 834 events in year 2022. Yeah, including symposium, seminars, receptions, as well as incubation programs, acceleration programs, pitching events, or matching events like this. Oh, we are quite happy and very excited to host today's events for more collaboration between UK and Japan. Please enjoy and utilize this opportunity for further collaboration. Thank you.
Cool. Thank you very much, Soyama-san. So now you understand more for the LinkJ. Good. And today we got 4 great speakers from Japanese ecosystems, and we nominated, and obviously they are the rising sun companies. And the first speaker will be Munemasa Sugimoto, CEO of Innopace. And if you can, yes, and they're doing a wireless powered closed loop neuromodulation systems. And I think Sugimoto is joining from Singapore today. And for your presentation, if you're ready, you can start.
Yes, thank you, Masashi-san, for your introduction. And let me show my screen. Okay, I think I will start. So thank you very much for this opportunity. And so this is Munemasa Sugimoto, the CEO of Innopace. So we are a Japanese medical device startup, and we treat chronic neurological disease. So our first target indication is overactive bladder, called OAB. So the patient are suffering from repeated urinary urgency and incontinence. So this problem is not only about when the patient have those symptoms. The fundamental problem is that patient keep feeling anxiety caused by the urinary incontinence, so which restricts the patient's productivity a lot. We can also imagine that this is not only about the physiological problem, but psychological problem. So if we consider this disease OAB from the economic point of view, so in the United States in 2007, the annual economic burden of overactive bladder OAB was huge, which was 66 billion US dollar. And also it was not only about the medical cost, but also the productivity loss, because the majority of the prevalence patients are still under 65, the active age. So this will tell us that many patients cannot go out or cannot work because of this disease. So if we think of the pathology of this disease, so this is not only about the bladder problem, but we need to understand that this disease is caused by the broken communication between brain and bladder. So here the key understanding is that the proper neural signal between brain and bladder is the key for bladder control. So the current standard of care, so there are millions of the prevalence patients. So in the United States, 33 million. In Japan, the 10 million. So those patients can start from the first line behavior therapy and the second line the medication therapy. But at the same time, we know that 30% of those patients become drug resistant, the refractory OAB. So they would need more advanced third line therapy, which is either sacral neuromodulation as a medical device solution or the Botox injection therapy. So our company is working on the sacral neuromodulation. The current unmet need is simple, just a lack of efficacy with current sacral neuromodulation therapy. So sacral neuromodulation, which is called SNM, so has been used in the clinical practice for more than 20 years. So we implant the pacemaker-like device in the body to give electrical stimulation to the target nerve, which is called the sacral nerve, to modulate nerve activity and manage symptoms. The current SNM device could only give abnormal constant stimulation. So we don't care whether we have the risk of incontinence or not. We don't care about the patient difference. We don't care about the disease progress. So we believe this is the primary reason why the current therapy efficacy is still limited. So if we look at the clinical evidence, despite of the sacral neuromodulation therapy, still the 60% of patients are not free from incontinence. And also the 8.5% of patients explant the device after implantation due to loss of efficacy. So we can see that there's a strong room to improve the therapy efficacy. So our solution is called the closed-loop sacral neuromodulation in combination of sensing and stimulation. So on top of the stimulation, our smart device can sense bladder afferent nerve activity and then give precision stimulation at the right strength and the right timing for patient. So this solution will achieve to suppress nerve habituation to provide long-term efficacy. And more importantly, this solution will facilitate neuroplasticity, which is the learning of brain, to restore brain-bladder communication. So we are aiming at higher chance of incontinence free for patients. So to support this concept, we have developed a functional prototype and worked on the disease model rat study for preclinical proof of concept POC. The objective here is to verify the feasibility of bladder afferent nerve activity sensing and preliminary efficacy evaluation of sacral neuromodulation. So these are some key data from our disease model rat study. So first about sensing. So our device senses bladder afferent nerve activity to predict symptoms. So here, if we look at this blue line, which is a nerve activity sensing measured by our own device. And for validation purpose, we also measure the bladder pressure. Understanding is nerve activity started more active before urination happened. So this will tell us that we can monitor the states of bladder in real time by sensing, and also we can predict the symptoms. With regard to the efficacy of precision stimulation, so our device give properly the precision stimulation to suppress enhancement of afferent nerve activity. And as a result, we could successfully extend the urinary interval. So on this slide, the urinary interval before we gave stimulation was around 4 minutes. But as we can see, once in the orange bar, once we gave the precision stimulation, the urinary interval was extended almost 3 times longer than the one before, around 11 to 12 minutes. So that we believe that this is a good preliminary result about the potential efficacy of precision stimulation. So regarding the competitive analysis, we have 2 competitors on the market. That's the sacral neuromodulation SNM, and we also have the Botox as a treatment competitor. But if we look at the incontinence-free, which was around the 20 to 40% range, so we are targeting much higher incontinence-free ratio supported by precision stimulation with sensing. And we are also expecting the longer duration of therapy efficacy, and we target 0% explant due to loss of efficacy. And as a result, that we can be superior to the from the economic point of view. The market opportunity, there's already $1 billion market globally by more than 50,000 annual implantation. But considering the much higher number of target patient population, we see the upside opportunity. Business model, we can leverage the existing reimbursement so that we sell our device to hospital, and the hospital use our device for therapy, and then the hospital can charge payers at around $10,000 to $15,000 per device. So the business roadmap, so we closed the pre-seed funding earlier this year to support the preclinical proof of concept study, and we are raising seed funding expected to close the quarter first next year to let us move to the clinical POC study. Upon the successful clinical POC, we are driving the product development non-clinical study and first-in-human study with fully implanted device. So we are targeting US, EU market approval in 2029 and Japan approval in 2030. Here's a management team. We have business and engineering background team driven by biodesign. We also have strong partners in Japan and globally. So as medical professionals, we are closely working with a series of the urology key opinion leaders and also neurosurgeon. We also have the data science, material science, brain science that expert, and we have the global business partner to support our activity. So we are currently looking for the $1 billion seed funding in March 2024. The key milestone will be to achieve clinical POC study and also the team building to activate global business development. So thank you for your attention.
Thank you, Sugio-san. Sorry for the inconveniences, happens, technical error. So I had a question, but maybe we can, for the time consideration, maybe we can do the Q&A maybe later on. Thank you very much. So next speaker is Kazuo Miyazaki, CEO of Micam Technologies. And they are doing, the title is Immortalized Monocyte Cells for MAT test, and they are doing a lot of activities outside Japan. So all for Miyazaki-san.
Okay, thank you for your introduction. Okay, I'd like to share the screen. Okay, good evening. Thank you for the introduction. I'm Kazuo Miyazaki of Micam Technologies. It's a great pleasure to attend the meeting and share with you our milk mat product. So in this presentation, the word A-milk will be mentioned. A-milk is a cell name. As you may know, monocyte are one of the white blood cell. When we create a new monocytic cell, I wanted to put a name with easy to remember. So I asked my scientists that what image when you listen white? So most of my staff answered that is milk, or like coffee with milk. So we decided to this cell name MYLC called milk. I would appreciate it if you could remember milk cell. Okay, start. First of all, let me tell you a little bit about myself. I was a medicinal chemist in a pharmaceutical company over 20 years. My life was triggered by my research on microbiology in India. When I lived in India, many colleagues suffer from malaria and dengue, and I totally recognize that the control of microbacteria is quite important. Since I had been working with stem cell technology in Japan, I began to think that stem cell technology could be used to solve that kind of several issues around immunology and microbiology. So after returning to Japan, I founded MyCan Technologies in 2016 to solve these issues. So MyCan is a biotech and a startup. Currently, we locate in Kyoto, Japan, and we are doing with 18 members. Okay, to begin with, I will talk about what is a problem in pyrogen test. I guess someone knows that rabbit fever test will be stopped by 2025 in EU because of animal welfare. But all drugs and vaccine requires testing for contamination prior to delivery. That is a problem. Okay, so currently, the contamination test will shift from the animal source to human-based test. But still, there is a problem. Okay, so I show you about the existence product for MAT test. So currently, many companies sell MAT test product. And if roughly separate product category, there are two types of products. One of them is a PBMC-based kit. I use blood. So there are many companies in these areas. However, as you know, since blood differs greatly from person to person, so that means it's difficult to get repeatability between batches. And the other one is also existence using a monocyte cell-based kits. A major product is using Monomax 6. But this cell established from the cancer patient and the chromosome number isn't 46. So when we create a new cell and new entry this market, our cells are high repeatability and also delivery from the human donor. So our technology and solutions. So I would like to show you our how to make AMX cells. We screen many PBMCs for IL-6 production after simulation by LPS and selected a high response PBMC. Then we separate a monocyte from the PBMC using MAX and several other techniques. The monocyte then did gene manipulation by our core technologies. Our core technology is immortalized gene transfection. Then to generate AMX cells. The comparison about the AMX cell and the monocyte from PBMC are shown below. AMX cells have almost the same properties as monocyte from PBMC. And also our core technology I would like to explain. Using the working cell bio for manufacturing, we increase the number of cells and prepare cell bios for MAT test. Currently, we are able to manufacture over 1,000 bios from just one bio within 2 weeks. That is our another core technologies. So both of the technologies use a comparison about the difference between PBMC and AMX. So PBMC products are prepared from the collection of highly reactive PBMC and mixed for 8 donors. And then manufacturing about the PBMC product for mat test. This product is in just 10 to 15% of monocyte because that rate is normal. But our milk cell is also using the high response PBMC and separate monocyte and then so immortalize. So that means our product consists almost 100% of monocyte. That is a difference between PBMC and milk. Okay, next I will show you about the competitive landscape. The milk mat product contains just 2,500 cells per well. Therefore, microscopic observation of the cell after the reaction shows this kind of figure. On the other hand, competitor use, for example, monomax 6 and PBMC needs 70,000 cells and 60,000 cells per well. We also sorted the LBS reactivity by OD values and observed that LOD, limit of detection, was low for both. But the angle of the rays, the AMX cells were quite higher. And also, I'll show you the presentation from Charles River Laboratories. Last month, the scientists from the Charles River Laboratories reported the comparison between a milk mat product and also existence product. So in this report, milk mat shows quite low LOD, limit of detections, 0.003 is quite lower than the competitive product. So I would like to explain about the development of the mat product currently situation. So currently, we are validation in the Charles River Laboratory in EU, and just test evaluation the customer sample now. And also, we are introduction of the other companies and optimization. So, and we will start to sell and service of the product from the 2025 January. Okay, so the request for the audience. So finally, request of the audience for pharmaceutical companies present, if decent. Please try to use MilkMat product for the pilot test, and we will support the implementation of MilkMat test. And for investor, we want to expand our business using this product in EU. So please invest me. And also, any audience, we would like to start our activities in Europe as soon as possible. Would you like to join us? So that's it. So thank you for your attentions.
Thank you, Miyazaki-san. That's very encouraging presentation. Thank you very much. I think this is quite strong message that the world's biggest CRO approved your product and you're ready to commercialize in the European UK market. Excellent presentation. Thank you very much. Right. Next speaker will be Erika Miwa, Business Development of Purec. The title is Highly Purified MSC for Cell Therapy Applications. Erika-san.
Hi. I'm sharing my screen.
Yep.
Can you see here? Can you see this?
Yes. Yeah, we can see. Thank you.
Thank you. Thank you for this great opportunity. I'm Erika Mia of Purec. So Purec is a biotech company with ultimate cell therapy using special MSC REC. So to illustrate our business, let me start with the challenges that we are facing. $2 billion. What do you think is this number? This is how much patients with hypophosphatasia have to pay for their drugs every year and their lifetime. So hypophosphatasia is a rare bone disease where the patient's body cannot make healthy bone and teeth. So where the current treatment does not cure completely this disease. So it's only delays the progression of these diseases. So MSC or mesenchymal stem cell could solve this problem. There was a successful case. There was a successful case in Japan with regular MSC. However, there was a problem. Regular MSC is not pure enough to be safe for the human body. In short, it is dangerous and non-effective. Our innovative product, REC, or rapidly expanding cells, overcome all the challenges regards to regular MSC. REC is highly purified. REC is highly purified and carefully selected MSC. Our founder, Dr. Yumi Matsuzaki, has developed the method for select the very best cells. There are two steps for producing REC. Firstly, we use cell sorter to choose candidate cell for REC from human bone marrow cells. And next, we culture the candidate cell one by one separately to find REC. No genetic modification and no special media, just by selection. REC is super elite MSC. It's superior to MSC in 3 ways. First, it does grow rapidly as its name. In 3 months, one single cell of REC multiplied to 1 trillion cells compared to 1 million in MSC. Secondly, it does grow rapidly. It does show clear differentiation, so especially into bone tissues. Lastly, REC can migrate freely throughout the body without any clocking. In other words, REC is low cost, and effective, and safe. REC is now under 2 clinical trials. The first one is for hypophosphatasia from earlier. It's a systemic injection. We will have only 3 cases because it is a rare disease. The next one is for spinal canal stenosis, a kind of disc herniation in the backbone area. This is a local injection of REC with special gel. We are already getting preclinical evidence in vivo about MSC's fundamental abilities, engraftment into injured tissues, and promotion for tissue regeneration. In an animal study of hypophosphatasia, we observed clear engraftment into mouse bone of REC provided from human osteoblast. Also, in another animal study of spinal canal stenosis, we observed many of regenerated disc cells in the damaged disc of sheep. Finally, let me touch our target market as well as our business model using REC. We are currently focusing just on 3 diseases related to bones like hypophosphatasia and spinal canal stenosis. There are already big market over $23 billion. Of course, we have other applications. However, as a small company, we just need a focus. And to achieve our business goal, the management team of REC is a set of quite experienced and balanced senior members. 3 are PhD and 2 are MD, including our very founder, Dr. Yumi Matsuzaki. Our CEO, he is now here, and Hide Takahashi has experience of drug development business both in US and Japan. So we are looking for partners and investors so we can together deliver our unique cell therapy using REG to more patients all over the world. Thank you very much.
Thank you very much, Erika-san. I think hyperphosphatemia has got a lot of patients. Yes, a lot of patients in the UK as well. So maybe you can encourage clinical study, etc., in this market. We're happy to help. Right. Thank you very much for your presentation. And the last one will be Kotaro Fujiyoshi, the head of CEO office of Synthetic Gestalt. So they are doing AI drug discovery services quite broadly. So Fujiyoshi-san, are you there?
Yes, I am. Can you see my screen currently?
Yes, yes, we can see your face as well.
Oh, so are you seeing the slide fully and not my list of people who've joined Zoom?
We can see full screen.
That's the full screen. Great. Yeah, okay. Then I'll get started. Thank you very much for this great opportunity. And yeah, I'll like to introduce our company and mainly our web service that we've launched a month ago. Because this is going to be of interest to medicinal chemists, I believe. So Synthetic Gestalt is basically a company made up mostly of machine learning engineers. We work in the interface between life science and AI to bring about, hopefully, new inventions that could help the world in many ways. So we do mainly two things, drug discovery and discovering enzymes. And yeah, I will mostly be focusing on drug discovery today. Within drug discovery, we've got two things that we're doing. So we are discovering novel small molecule compounds that could become therapeutic in an in-house discovery. And in a web service, we're offering internally developed machine learning models to, one, predict compound issues, and two, propose compounds with those issues resolved. I'm going to be talking mostly about the web service. Right. So what does our web service do? It is my understanding that some medicinal chemists suffer from a lack of direction in structural optimization when they get compounds from HDS. And basically what our service does is it allows the users to easily browse through new ideas, new chemical structures that, as if you were asking ChatGPT, it says in this slide, and yeah, get the information, the sort of prediction values from a machine learning models to decide on the pathway of overall direction of the research. Yeah, so it offers two functions, prediction and proposal. Prediction is basically, the letters are a little bit tiny in this slide, but when you input the compounds into the service predicts these ADMET and physical chemical values as such on the x-axis, and on the y-axis you have these compounds. And basically, in this first compound, let's say, the machine learning model is saying for CYP1A2, the target, it says it's got 87.1% confidence of the inhibition of this target being more than 30 micromolars. So basically it's got a bit of a mid confidence in terms of it being bad in terms of this metric, and it predicts all a lot of metrics for physical chemicals and ADMET, up to 54 parameters. And overall, it boasts accuracy of over 90%, I've been told, achieved on actual experimental data, which is quite impressive, I believe. And the second function of this is basically when you want to develop a particular compound out of your list, it proposes the compounds with those problems identified in this first part solved. basically what it does is it searches the immediate chemical proximal compounds, so with a small distance to the original compounds, basically, that clears the parameters that user specifically wants to clear. So for the first compound, let's say you want to have a green traffic light. These lights are basically traffic lights. This yellow means not so good. For this first compound, let's say you wanted to clear CYP182 into green, and then you specify that, and then the model basically fetches from a long list of compounds derived from easily synthesizable lists from Enamine and Wuxi Aptec. It suggests a compound that is in the vicinity and is predicted to clear the threshold that the user sets. So basically, it's easy to use is one of the great pluses of this service. You just input the smiles. We're gonna work on more input methods in the future, but we just work with smiles and CSV files at the moment. Anyways, you input the compound structure, wait a few seconds, and you can do predictions and get new compound proposals straight away is the idea of the app. Yeah, this pretty much covers the what I've already said, but if you click on each of the compounds, it just shows the the basic information of compounds and the predictive values for each of the parameters. HERG, CYP, log-D, log-S, Kagotu PAP, and a few other metrics. Up to 54 is predicted. So yeah, the fast and easy is the proposal that we're making with this product, and the user can easily set these thresholds. For example, just clicking on this particular button would set the CYP do D6 to more than 90%, and just click apply, and then get compound proposals. And yeah, so why we developed this product is that we've got these stocks of machine learning models that within the company. And we thought there might be a sort of chance for other medicinal chemists in other companies to use this particular set of models that we've made to balance between all these risks that they have to manage in their day-to-day job. And that basically wraps up the product that we are currently offering for this. In other parts of our business, we're doing in-house drug discovery. I haven't got a lot of slides for the in-house drug discovery, I'm afraid, but we've got a joint project with DWTI going on in this respect. And I don't believe any other companies in Japan that does AI drug discovery has licensed out a compound yet. But we are working on it. We've got an internal pipeline that is running, and we might get to that stage within half a year or so. So yeah, we are working on that front as well. Looking slightly away from drug discovery, we're also doing a solution business where we solve other companies' problems using biotechnology, basically. And what we do essentially is predict novel enzyme functions. So basically, for a given set of amino acid sequences, we try to predict what the function of the amino acid sequences is in the in terms of enzymatic function. So we've not, for example, we've identified 4 novel PET degrading enzymes, PET being the plastic, PET, and we are pursuing businesses with other companies like Tokyo Gas in trying to find a novel enzyme, for example. And we provide this enzyme function identifying capabilities for other companies. And yeah, we basically take 1 month to hear out the needs and a few months to implement. And so yeah, if anyone listening is interested in this sort of service, then yeah, please let us know. So yeah, overall, I just wanted to introduce what we do. If anyone is interested in using any of our services, then we'd be very happy to know. And also, we are doing a bit of a fundraising round in the coming months. So if any investor is interested, please let us know, and we'll be very happy to start talking. So I can go on about our company, but I think there's been a few mishaps. So I think I'll just leave my presentation here. And yeah, if anyone has any questions, yeah, please let us know.
Well, thank you very much for your science, quite good presentation. Quick question. So you also looking for fundraising. Can you tell us more in particular?
Okay, so we have finished Series A2 fundraising in 2021, I believe, and we've raised $14 million so far. We're doing a Series B fundraising this time around to raise funds for further taking internal pipeline to the next stage, and also developing our the web service that I've just shared with you. And we have a few scenarios in mind, but the main scenario, I think, is we're looking for a lead investor that can set the price range for us. And we are looking at about ticket size of $3 million or more. So yeah, lead investor or investor interested in ticket size of $3 million or more in investing in internal drug discovery and web service.
Excellent. Thank you very much. Because your company has got legal entity in the UK, so maybe it's much easier to access.
Yeah, about that. Sorry, we were so a little bit about our company. We were founded in the UK and Japan, and the UK was the parent company till quite recently, but we've gone through an inversion process where Japan is the main parent organization currently. However, we do have the British Business Bank in one of our shareholders. So yeah, if that can act as a sort of relief for potential UK investors, then the information is also there.
Sure. Thank you very much. Thank you.
Thank you.
Good. Thank you for all the speakers. That's excellent presentation. We still got 10 minutes, even though we got technical difficulty. So maybe, would you mind if I can have your faces, all the speakers? I have a quick questions. Starting from Sugimoto-san, Innovbase.
Yes.
You still there? Yes. So your wish is for fundraise for 1 million for the seed fund. Can you explain more, and where will be the UK target, for example, for your device?
Yes, so currently we are raising the $1 billion, and half of the 50% are almost confirmed with our current investors. So we are still looking for the $500,000. And we are currently communicating with some Japanese investors, but at the same time, because Europe market and the US market are quite important for us for the commercialization. So we are looking for the Europe-based and the US-based investors, including the angel investor, not only to help us financially, but also to help us strengthen our team and the networking for the future growth.
Thank you. Are you interested in doing like a clinical study in the UK or in Europe?
Yes, so there will be the series of the clinical studies. So that I think that we will consider the clinical studies at some point in Europe as well. And I'm exploring the opportunity in the UK as well.
Cool, thank you. Because in which case you will need to raise more funds, obviously.
Yeah.
So yes, good luck, and thank you very much for answering.
Thank you.
Thank you. Second question is Miyazaki-san, Mike-san.
Oh, yes.
Yes, so I'm quite interested in your success in collaboration with the Charles River. What is your commercialization plan? And are you looking for like allocating some space or demo lab in Europe or UK?
Yes. So we will have a plan to start to sell from the 2025 January. So we are looking for the test use customers, a potential customer. We are looking for a potential customer. Also, so the like a pharmaceutical company or the CRO or medical device company. So and also so we have to implement how to evaluate. So that's why also we are looking for some of the demonstration laboratories. So if someone want to know about more in detail our math test, so I would like to show you.
Sure, thank you very much. Yes, so January 2025, it's coming soon.
Yes, quite coming
yes.
soon.
And good luck. Thank you very much. Purek, maybe Takahashi-san or
Okay, why
Airika-san.
not?
What
Yeah.
is your strategy for UK collaborator? Do you want to collaborate with the UK universities or hospitals?
Yeah, definitely. I already had several contacts at UK universities. There are very, talented, researchers in the UK, especially for the hypospatasia clinical trials. So I already have a contact at several universities, but I want to expand network a little more. And also, we may need some facility for the production in the UK. So the CDMO is another target, which is also, I made some contacts in several cities. So UK must be my favorite area. So I want to establish my entity there, but not yet. So yeah. Probably Erica would be the representative in the UK operation, probably. So, okay, yeah.
Okay, so new HR announcement. Right, I think having like a CDMO function in the UK is quite maybe promising ideas. Great, thank you very much indeed. And for the time consideration, maybe I will send my microphone back to One Nucleus team.
Yeah, does if no one else has any more questions, just want to say thank you everyone for their presentations, and wherever you are in the world, I hope you have a good rest of your day.
Right, thank you very much all.
Thank you.
Thank you.
Bye.
Okay, thank
Bye-bye.
you.
Thank you. Bye.